Amino acids, proteins, and DNA. Definition of amino acid has a basic amino group NH2 and an acidic carboxyl group COOH, therefore making amino acids amphoteric. They have acidic and basic properties. Amino acid basic formula CH NH2 C double bond OH, and this is the only thing that changes to R, the organic side chain. Now, all amino acids are chiral molecules. False, except for glycine, because glycine, this organic side chain, is a hydrogen. Amino acids have an amino group and a carboxyl group. Amino acids can exist as zwitterions. Say a key fact about amino acids: they are chiral molecules, yes, except for glycine. Carbon has 4 different groups attached. Therefore, a solution of a single amino acid enantiomer will rotate plane polarized light. And you need to, be very specific with what you're saying, because in the mark scheme they're looking for that. A solution of a single amino acid enantiomer will rotate plane polarized light. Definition of zwitterion: a dipolar ion has both a positive and a negative charge in different parts of the molecule. Positive charge and negative charge in different parts of the molecule. They only exist near amino acids. They only exist near amino acids at isoelectric point. Isoelectric point, the pH, where the average overall charge on the amino acid is 0. pH, or the average overall charge on the amino acid, is 0, and it depends on R groups. Therefore, isoelectric point is different for different amino acids because it depends on R groups. Low pH conditions more acidic than isoelectric point. COO- group gains a H+, NH3 plus CRH, C double bond OH. Zwitterion, CRH, NH3 plus C double bond O- at isoelectric point. Both carboxyl group and amino group are ionized. At high pH are CH, NH2, C double bond O-. Conditions more alkaline at isoelectric point. NH3 plus group loses a H+. Proteins are polyamides. Monomers are joined by amide groups in proteins called peptide links. Proteins are made of amino acids joined together by peptide links. Proteins are condensation polymers of amino acids. And this is what I mean. So you got 2 amino acids, CRH, NH2COH, and another amino acid with different organic side chain, CHR-NH2COH, OH and a H here, reversible reaction, condensation to go here, hydrolysis to reverse the reaction conditions, and then you form a peptide link plus your H2O. Say the 2 conditions to break a protein into its individual amino acids. Basically, what I should have said, say the 2 conditions to hydrolyze a protein, which is breaking a protein into its individual amino acids. That is just helping with your understanding. What's happening in the hydrolysis, you're breaking a protein back into its original amino acids, individual amino acids. Add hot aqueous 6 meters of hydrochloric acid. Heat the mixture under reflux for 24 hours. Primary structure, the sequence of amino acids in a long chain that make up the protein, the polypeptide chain. Secondary structure, peptide links form hydrogen bonds with each other, therefore chain isn't a straight line. Shape of chain is called a secondary structure. Say the most common secondary structure, spiral, called alpha helix, or beef pleated sheet. This is a layer of protein folded like a concertina. Tertiary structure, the chain of amino acids is coiled and folded in a characteristic way that identifies a protein, plus extra bonds formed between different parts of polypeptide chain, giving the protein's 3D shape, which is its tertiary structure. Alpha helix chain is coiled into a tertiary structure. Secondary and tertiary structures of proteins are formed by intermolecular forces causing amino acid chains to fold/twist. Intermolecular forces are vital because the 3D shape of a protein is vital to how it functions. Hydrogen bonding, should know from year 1, rebonding, exists between polar groups, OH and H2, stabilize both the secondary and the tertiary structure of proteins. Amino acid cysteine contains a thiol group, and that's really important because amino acid cysteine contains a thiol group -SH. Therefore, the thiol groups and different cysteine residues, can lose H atoms and join together, forming a disulfide bond, a sulfur-sulfur bond, helping to stabilize tertiary structure. Substrates are molecules that enzymes act on to speed up reactions. Enzymes, proteins that act as biological catalysts. They catalyze every metabolic reaction in the bodies of living organisms. Enzymes are proteins, and some have non-protein components. Every enzyme has an active site, part that the substrate fits into so it can interact with the enzyme. Active site is 3-dimensional, part of the tertiary structure of protein. Enzymes are made up of amino acids, therefore they contain chiral centers. made of amino acids that contain chiral centers. Active sites are stereo-specific. They only work on one enantiomer of a substance. The other enantiomer won't fit properly in active site, so enzyme can't work on it. Molecules that have a similar shape to the substrate act as enzyme inhibitors, competing with the substrate to bond to the active site, blocking the active site so no substrate fits. State 2 key facts about inhibition. How much inhibition happens depends on the relative concentrations of inhibitor/substrate. Amount of inhibition is affected by how strongly the inhibitor bonds to the active site. State 3 ways of finding a drug molecule fitting into the active sites. Now, this is a part of the specification where it's more like how scientists trial and error using computers, etc. Trial and error scientists carry experiments using lots of compounds to see if they work as inhibitors for a particular enzyme. Adopt any that work and try to improve. Pretty self-explanatory, right? It's trial and error. You're doing a bunch of compounds, see if they work as inhibitors. If they do, particular enzyme, you adapt any that work and then try to improve them. Maybe making it faster. Using computers to model the shape of an enzyme active site. So yeah, that's what we're using computers to model the shape of the enzyme's active site and then therefore predict how well potential drug molecules would interact with it. Therefore, this means that scientists can quickly examine hundreds of molecules to look for the right shape before synthesizing/testing things in laboratory. DNA, polymer of nucleotides made up from lots of monomers called nucleotides. Nucleotides. A polymer of nucleotides made up from lots of monomers called nucleotides. Nucleotides join together to form a polynucleotide chain. Covalent bonds form between a phosphate group of one nucleotide and sugar of another. This is what makes the sugar-phosphate backbone of the chain. DNA forms by condensation, polymerization, and what happens is a molecule of water is lost and a covalent phosphodiester. A molecule of water is lost and a covalent phosphodiester bond is formed. DNA formed by condensation polymerization. Sugar phosphate backbone of DNA is formed by condensation polymerization. DNA is made up of two polynucleotide strands, double helix structure. Two strands spiral together to form a double helix structure, which is held together by hydrogen bonds between the bases. The two strands of DNA are complementary. Adenine always bonds with thymine. Guanine always bonds with cytosine, ATGC. They match up, so whenever there's an adenine base on a strand, there will be a thymine base on the other, and whenever there's a guanine base on one strand, there'll be a cytosine base on the other. And that's really it for amino acid protein DNA. There is a bit more on cisplatin, but that is what I'm probably going to create into YouTube shorts, because that's very like a lot of how do I say this, a lot of obviously information. I want to put that as my YouTube shorts on cisplatin, and then that's it for amino acids, proteins and DNA.